Hello, everybody. Welcome. Welcome to our fourth session for the Reader's Talk Green. This time we'll speak about genetic diversity and responsible breeding. I would like to welcome our EFFAB members. We are just entering a new CAP today for sustainable and responsible agriculture, preserving our great planet and food security. And I welcome also, of course, our guests. It's very important for us, members of EFFAB, to share with you our thoughts on diversity, genetic diversity. So I will let the floor to Anna and her team in EFFAB, who will do a very great job for organizing these breeder to grain sessions. Thank you, Anna.
Thank you very much, Pascale, for this welcome, and welcome to everyone today for this fourth session, as you said, on animal genetic diversity. So yes, let's start just to show you the team behind the screens to make this session again a success, and about EFFAB and FABRE-TP members and EFFAB and FABRE-TP missions. So as you know, we are the European Forum of Farm Animal Breeders, and FABRE-TP is the farm animal breeding and reproduction technology platform active at the European level on policy, but also on research. We are also involved in European projects. We are now, we have currently 8 projects to starting still this year. So let me remind you the house rules. Please use the Q&A box for questions. Only use the chat for technical issues or this kind of things. But for questions for speakers, please use the Q&A box. We will address questions at the end of the presentations to start the panel discussion. When you have started your session, you have seen that the webinar is recorded. It will be shared in our social media and website later. We will provide also a summary. So as said, you can ask your questions in the Q&A box. The moderator, Chala, and me will collect them and present them to the end of the presentations. You can also take part in polls with a Slido tool. You have the code here, V319. We have only 2 questions today about the topic of genetic diversity and the challenge for animal breeders managing genetic diversity. So the context of this session today is, of course, again, the European Green Deal and the strategies from farm to fork and biodiversity for 2030. I would like just to show you how animal breeding is important. It's a great driver for more sustainable food production, as we are, as this is foreseen in the objectives of the European Green Deal. And around the Code FABRE, what is the Code of Good Practice for Responsible and Balanced Breeding, I just want, our webinars are around this Code FABRE at the pillars for sustainable breeding, and one of these pillars is genetic diversity. So making sure that there is a higher level of genetic diversity within population, but also that our breeding organizations are aware of the necessity to maintain this genetic diversity. So let me go through the program to start as soon as possible. We have made the welcome and the opening, and we are now going to welcome Paul Boettcher, who was not able to come. Go back, please. Who was not able to come today because he's in another meeting, and he's from FAO and from the FAO, the Animal Genetic Resources Department or unit, and he will make the first keynote today to draft the global level of animal genetic resources. And after we'll go down to the European level. So with Sipke, Rachel, and Pierrick. In the panel discussion, we will have 3 more panelists. So Paul is not able to join us. So his colleague at FAO Fisheries, Graham Mair, will join us. And 2 more panelists, Juan María Gallardo from FEAGAS, a Spanish organization, and Waltraud Kugler from SAFE Foundation will join later. So let's start with the first presentation. Thanks, Isabella. Will be a video of around 6-7 minutes from Paul, who is in charge of animal genetic resources at FAO. So let's launch the video here.
Good day, everyone. I'm Paul Boettcher, Animal Production Officer with the Food and Agriculture Organization of the United Nations, better known as the FAO. Today I'm going to talk to you about FAO's work on animal genetic resources and animal genetic diversity. International cooperation is required to ensure continued access to a sufficient and healthy food. Countries differ vastly in their capacity for production of food, and natural agriculture consumes immense natural resources. The United Nations therefore has a logical role to play in the international food system. The Food and Agriculture Organization is the specialized agency of the United Nations that leads international efforts to defeat hunger, and it was founded in October of 1945. So how does FAO get the job done? Its work is done through 5 different means: putting information into reach, developing capacities within countries, providing policy support, providing a neutral meeting place for nations, and sharing know-how, skills, and expertise. So why do animal genetic resources fit into this picture? Well, animal genetic resources and diversity are an essential part of the biological basis for world food security. 1 billion people rely directly on livestock for a major proportion of their livelihood, and billions more rely on livestock products as part of their diets. A diverse resource base is critical to eradicate food hunger. Livestock genetic diversity allows for adaptation to current and future environmental constraints, such as climate change, and also serves as the raw material for breeders to make genetic improvement. Although individual animals are owned by individual people and countries are responsible for their own breeds, this genetic diversity is considered an international public good, and therefore FAO has a logical role in the global coordination of its management. FAO member countries developed an international policy for genetic resources called the Global Plan of Action for Animal Genetic Resources. This Global Plan of Action includes 23 strategic priorities for action aimed at addressing future challenges to the livestock sector. These priorities are assigned to different strategic priority areas, the first of which is characterization, inventory, and monitoring. The second is sustainable use and development. The third is conservation, and the fourth is policies, institutions, and capacity building. The main responsibility for implementing the Global Plan of Action relies on national governments. This is because, I had mentioned before, the breeds are sovereign to each country. There is, however, a need for international cooperation, and particularly to support developing countries. And this support is provided by international organizations, including FAO. So FAO provides technical support to their countries. In addition, FAO is responsible for monitoring the implementation of the Global Plan of Action. So I'll now explain how FAO contributes to this, relying on each of the different 5 areas of work, or modes of work for FAO. With regard to putting information within reach, every 10 years, FAO assesses the implementation of the Global Plan of Action and prepares state-of-the-world reports on the status of animal genetic resources. FAO also maintains a domestic animal diversity system, better known as DADIS, which is a database of breed descriptions and population data provided by countries. This information and data serves as a source of data for monitoring indicators 2.51 and 2.52 of the Sustainable Development Goals, which monitor the status of genetic resources ex situ and in situ, respectively. FAO also hosts DataNet, which is an online discussion group with more than 3,000 members and publishes scientific articles and technical reports. With regard to developing capacity, FAO organizes training courses on the national and regional levels and also contributes lectures to partners' training events. In addition, FAO develops guidelines and other training materials for these events and for direct use by individuals. With regard to providing policy support, FAO assists countries to develop national strategies and action plans for animal genetic resources, which emphasize the priorities in the GPA, which they consider most important for their country. FAO provides a neutral meeting place for nations. FAO provides secretaries for the intergovernmental bodies on agrobiodiversity, including the Commission on Genetic Resources for Food and Agriculture, which is a policy-making group, and the Individual Technical Working Group on Animal Genetic Resources, which advises the commissions on technical and scientific issues. These groups meet every 2 years to review the status of animal genetic resources and deliberate on actions to be taken. Finally, FAO supports through sharing know-how, skills, and expertise. FAO implements multinational and national projects, including through FAO's Technical Cooperation Department, through donor-funded projects, and through the funding strategy for the implementation of the Global Plan of Action for Animal Genetic Resources. And finally, FAO also provides staff to support technical expertise for other intergovernmental agencies, such as the World Bank. So with that, I'd like to say that I hope this has provided you some information on FAO's work on animal genetic resources, and I'd like to thank you for your attention.
Okay. So yeah, we thank Paul for the video, but he's not with us today. So now it is time to go to the second presentation. Will be done by Sipke Hemstra, who is the chair of the steering committee of the European Regional Focal Point for Animal Genetic Resources. He's also working as director of the Center of the Genetic Resources in Wageningen on the Dutch level. And we should be able to see him. I can see you.
Thank you, Anna, for your introduction. Thanks to the organizers for inviting me to talk in this webinar. So today I will talk on behalf of the European Regional Focal Point that I'm currently chairing. I will present the Generous Bridge project and the process we are currently in, developing an integrated European genetic resources strategy for plant, animal, and forest genetic resources in the context of the EU Green Deal and other relevant European and international obligations and policies. After introducing the integrated genetic resources strategy for plant, animal, and forest genetic resources, I will focus a bit more on the development of the domain-specific animal genetic resources strategy that we are currently developing in the ERFPE network. So about 1 year ago, the European Commission announced the European Green Deal with the main aim to make the EU economy sustainable, to move to a clean and circular economy, and to restore biodiversity. The Green Deal also implies a transition in our food system and in agriculture, and that will require a collective approach, including a diversity of actors. It is obvious that optimal use of genetic resources, genetic diversity, is a key component for the transition towards sustainable and resilient agriculture and food systems in the context of the Green Deal, and to meet international obligations and agreements such as the Sustainable Development Goals and the commitments in the context of the FAO and the Convention on Biological Diversity. In the Farm to Fork strategy and the biodiversity strategy under the Green Deal, plant and animal genetic resources are mentioned, but not very prominent. Following the request of the European Parliament and a preparatory action on genetic resources of the European Commission, the 3 European networks on plant, animal, and forest genetic resources, ECPGR for plants, EU4Gen for forest, and ERFP for farm animals, submitted a project proposal, Generous Bridge, a coordination and support action. And the aim of this proposal, Generous Bridge, was to develop a European genetic resources strategy, an integrated strategy for plant, animal, and forest genetic resources. More recently, in October 2020, EU Council conclusions on the Farm to Fork strategy also highlighted the importance of ensuring the protection, restoration, and sustainable use of genetic resources of plants and animals. And the Council invited the Commission to present an EU strategy for genetic resources for aquaculture, forest, and agriculture that is based on the work of the FAO Commission on Genetic Resources. And the Generous Bridge project has now developed such a strategy for genetic resources for further consideration by the EU, individual countries, and the European network programs. So let me first address the question why it is important to develop this European strategy for genetic resources. First of all, it's very relevant to demonstrate the importance of genetic resources for food and agriculture in the context of major European and global challenges, in particular the EU Green Deal. There is currently also a lack of coordination of genetic resources related policy development. There's clearly a need to provide a European policy framework for more effective safeguarding and sustainable use of genetic resources for agriculture and forestry. And finally, with this integrated strategy, we also see opportunities to strengthen the synergies between plant, animal, and forest genetic resources domains and policies. Future adoption of this strategy and implementation of the European Genetic Resources Strategy should minimize the loss of genetic resources, minimize the loss of genetic diversity between and within species, breeds, and varieties. The strategy should support diversification and innovation in agriculture and forestry and build resilience in agriculture and forestry, keeping in mind the multiple challenges that we are facing. Genetic diversity between and within species, breeds, and varieties is the key for enabling an adaptation to changes in climate, production systems, and consumer preferences. So this slide shows the outline of the integrated European Genetic Resources Strategy. Following the rationale and the need for urgent action in section 1, we describe the main objectives and key commitments of the strategy in section 2, strengthening and widening the conservation and sustainable use. In this section, we discuss the conservation, sustainable use, diversification, characterization, monitoring, and information management goals. Section 3 is about enabling the suggested transformative change. In this section, we discuss the establishment of an appropriate European policy and legal framework, the need to increase institutional and human capacities, enhancing awareness of the role and values of genetic resources, mobilizing funds, and the opportunities for joining forces between the different domains. The European Genetic Resources Strategy that we are developing will be finalized in September of this year, following several stakeholder consultations in the past year. And the dissemination event is planned in November of this year in Brussels, where the strategy will be presented. So the integrated overarching genetic resources strategy is about to be finalized. In line with this overarching strategy, ERFPE, the European Network for Animal Genetic Resources, and also the plant and forest networks are currently developing also their own domain-specific strategies. And the Animal Genetic Resources Strategy will be more specific on issues particularly relevant for animal genetic resources and the livestock sector. So the term animal genetic resources refers to the genetic base that is needed today and in the future to be able to adapt to changes in production systems, climate, or demands of consumers and society. It is about the genetic diversity between and within species and breeds, including local breeds and more widely used mainstream or transboundary breeds. And genetic resources include live breeding populations, but also reproductive material and DNA. So in the first pie chart at the top of this slide that is derived from FAO data, we see that there's a very large number of breeds defined as local breeds, occurring in only one country. And the number of regional or global transboundary breeds is relatively small to the number of local breeds. In the second pie chart, that is the Sustainable Development Goal Indicator 2.5.2, also based on FAO data, shows that the majority of the local breeds in Europe that has a known population size can be considered as at risk of extinction. And the risk status of breeds is based on the population size of the breeding population, but we also know that many breeds also have a very low effective population size, a low within breed genetic diversity. The major reason for many local breeds being at risk of extinction is the gradual replacement of local breeds by specialized high input, high output breeds in the past decades. And at the same time, we know that the diversity of breeds and livestock systems provide a variety of ecosystem services. The Millennium Ecosystem Assessment identified 4 categories of ecosystem services. Provisioning services are the products obtained from ecosystems, for example, food and raw material of various kinds. And examples of regulating services include regulation of the climate, air and water quality, or flood regulation. The socio-cultural value or recreational value of livestock systems and breeds are recognized as cultural services. Supporting services or habitat services are services necessary for the production of all other ecosystem services. Examples are soil formation or pollination. So we can conclude from this that the diversity of livestock systems, including the breeds used in these systems, could be better recognized and valued for the range of ecosystem services that they provide. There's a clear need for policies and strategies to safeguard the farm animal genetic diversity between and within species and breeds for current and future use, and to recognize the different values and ecosystem services associated with specific livestock systems and breeds. And the FAO Global Plan of Action that was presented already is the agreed global framework for this. I cannot go into details in this short presentation, but I want to highlight some key elements in the European Animal Genetic Resources Strategy that is currently under development. First, in the farm animal domain, we consider conservation by utilization as the main conservation strategy. The role of breeders, breed societies, and breeding industry is to implement effective and tailored breeding programs for a diversity of systems. And breeding programs should always find a balance between genetic improvement and maintaining genetic diversity. In addition to the management of genetic diversity in breeding programs, it is recommended to implement a complementary gene bank strategy for all breeds and breeding programs for long-term conservation, and as an insurance for unforeseen events. We should not wait establishing gene bank collections until breeds are already at risk. It's also important to enhance our knowledge about characteristics, performance, and values of breeds in a range of production systems. And at the policy level, better coordination is needed. In particular, better integration of conservation and sustainable use of animal genetic resources goals in different, often already existing policies and legislation, such as the Common Agricultural Policy, breeding legislation, animal health legislation. And finally, a collective approach is needed to reach our short-term and long-term objectives, involving public authorities, animal breeders, and other private sector actors across the value chain, NGOs, academics, and consumers. One example of important public-private collaboration is the establishment and maintenance of Genebanks for long-term conservation purposes. As shown in this slide, Genebanks are important repositories of farm animal genetic diversity that can play an important role in dynamic breed management, not only as a backup for re-establishment of breeding populations that may become extinct. There are many examples of the use of Genebanks to support the conservation and breeding programs of endangered breeds. And Genebanks are also relevant as a resource base for scientific research, for monitoring of genetic diversity, or for building reference populations. The ERFPE Network, the European Regional Focal Point, network of national coordinators, started the development of a European network of Genebanks called IGENA. And IGENA member Genebanks are officially recognized Genebanks by the national governments for the purpose of long-term conservation and sustainable use of animal genetic resources. To conclude this presentation, I would like to highlight that breeders and farmers are actually the main actors in the European strategy for the conservation and sustainable use of animal genetic resources, and that there are many opportunities for further developing private-public partnerships and collaboration to reach our common goals. ERFPE, the network of national coordinators for animal genetic resources, will continue its work in the next years to promote conservation strategies and sustainable breeding programs, to monitor trends in genetic diversity, and to increase awareness about the roles and values of animal genetic resources. Our aim is to collaborate with all relevant actors and stakeholders to reach our common goals. Finally, I would like to acknowledge the ERFPE Task Force that is involved in development of the European Genetic Resources Strategy and the domain-specific strategy. And I would like to acknowledge the funding provided by the EU Horizon 2020 program for developing the integrated strategy for plant, animal, and forest genetic resources. Thank you for your attention.
Thanks, Sipke. Thank you very much. I hope you enjoy the presentation because there was a lot of things in. I mean, I was aware about a lot of things because we have been involved also in drafting this animal genetics resources strategy. Not only animal, sorry, it's also for forestry and plants. But there was a lot of information in, so don't hesitate to ask questions in the QA box. Before continuing with the presentation with Rachel, we have a first question for the audience to try to engage you more with the panelists. So the question is, do you think you can go through a Slido, connect with a Slido and the code V319? And the question is, do you think the importance of the native local breeds should increase in commercial animal breeding programs in the coming 10 years? Well, we are going to give some minutes to the audience to respond to the question. And well, in the Slido is also always quite interesting to see how the answers are moving. Okay, nobody responding now. We are now only, yeah, people is connecting. Okay, 34 answers, 36 is going up. Rachel should start at 2:05. So yeah, let's give 1 minute or 2 minutes more to answer to the audience. Okay, I think it's not moving a lot. So yeah, should, but only 40 answers. So it's perhaps not representative of 230 attendees. So I hope that for the next Slido after the two presentations, yeah, we will have more response. So now is Rachelhaugen from Cobb, a poultry breeder member of EFFAB, who is going to take the floor to go more in details or into the practice on how to implement genetic diversity. I'm sorry. How to maintain genetic diversity within breeding programs, within breeding association. So Rachel, I'm sorry, I have missed my notes. Rachel is senior. Hello, Rachel. We can see you. Is senior director of genetics at Cobb, as I said, a poultry member of EFFAB, and she has a strong background in genetics, with several positions in research previous to her position in Cobb. So the floor is yours. I think we can hear you too.
Excellent.
And yeah, exactly. And you can share your presentation. I'll stop talking.
Okay. Thank you. Well, I appreciate the opportunity to talk with you today. So as was mentioned just a little earlier, I'm here representing Cobb Bantress as a poultry breeder. For those of you that are not aware, Cobb provides the genetics of about 50% of the global poultry broiler market. So we have a great responsibility to maintain production, to maintain performance over the next future of our industry. So we take genetic diversity very, very seriously. If you could go to the next slide. So what I'm going to go through today is really just a brief overview of the needs of the future. And it's really just a list that I put together out of thinking about why is genetic diversity important. And then I'm going to talk a little bit about the genetic diversity that we have in broilers or in broiler genetics. And lastly, and actually mostly, I'm going to talk about the new technologies that we have developed in collaboration with the Roslin Institute to maintain some diversity for future needs. Next slide. So future needs. So, you know, when you sit down and you think, well, why is genetic diversity important? You know, there's a lot of studies out there looking at why is genetic diversity important. Well, why is genetic diversity important to a poultry company, where poultry companies push to produce a product that is sustainable, it's efficient, it's cost effective, and it, most of all, that it produces a source of cheap animal protein for global consumption. But there are several things that come to mind when thinking about why this is important. When you think about changing global markets, climate change, of course, we know that broiler production, our broilers have a range of temperatures that they perform well at, but you know, withstanding heat and withstanding cold is difficult for them in some instances. We know that there's pathogen evolution. We know that there's pathogen migration. And we know that in the future that nutrition source, or what we call responsible resource use, is going to be an important aspect for future animal production needs. So given all that, it would be negligent of us to assume that our current commercial products are going to be able to maintain their performance given all these factors. So if you go to the next slide, you know, we have 4 main products out there in the market, 2 female, 2 male products. And this is the majority of our sales globally. We have the Cobb 500 and the Cobb 700. Now, when we look at the genetics or the DNA profile of these birds compared to each other, we have millions of genetic variants that exist within and between the lines that are created to create these products, remembering that these products are a crossbred. So we do a very good job of maintaining as much diversity as we can while still making a product that performs well in as many global marketplaces as we can. If you go to the next slide, the next slide shows also a range of birds that are, we don't call, they're not our commercial lines, we call them actually our zoo. These are genetic lines that we keep because we know that at one day these are going to be important. We know that once we get into investigating the genetics of these lines and the performance, the unique performance characteristics some of these lines produce or contain, we will want to identify genetic elements within these lines and introduce them into our commercial lines, or ramp up production of these lines to be included in the crossbred production of a broiler. So we go to great lengths to maintain genetic diversity as a broiler breeder. If you go to the next slide, there are many options to maintain genetic diversity, some better than others. Of course, you know, the best way to maintain genetic diversity is maintain live flocks. It's really maintaining biodiversity, really. Maintaining live flocks of these chickens is an expensive task. It's a continuous task. And to be honest, it's at risk of disease outbreak. What happens if we have, you know, limited flocks of these zoo animals, as we call them, these zoo chickens, and we have a disease outbreak come through and we have to cull large populations of these unique genetics? Well, that puts us at risk of not having that genetic diversity available to us. We can also freeze germplasm. There's been a lot of efforts around the world over the last decades to freeze semen, ovaries and testes, and then reintroduce those tissue supplies back into chickens, so that we can recreate populations of different lines, different breeds of birds. There is a lot of success here, but the success is not really at the level of being able to recreate a large flock that would be necessary for to supply global chickens, or chickens globally, excuse me. Freezing PGCs and being able to recreate a population from those PGCs, and PGCs are primordial germ cells, is really the technology that we have spent a lot of time and money on over the last several years to develop a system that's high throughput enough that a company like Cobb can reintroduce a flock if necessary. So if you go to the next slide, PGC Technology really is a technology that allows us to take the primordial germ cells. Now, primordial germ cells are represented in early embryos, very early embryos. They're available to collect, and at a certain day of development, embryo development, you can actually retrieve the primordial germ cells from an egg, from an embryo in an egg, and utilize those PGCs for any task that you require. Those PGCs end up at day 5 forming the gonadal ridge and being responsible for the germplasm in the testes and the ovaries. So if you go to the next slide, this is actually a video. We can actually, when I say we, it's the royal we. I haven't done any of this. This is all done by the Roslin Institute. In fact, Mike McGrew, who is on this conference, is responsible for this work, and our collaborators. This is a video of the collection of PGCs from an early embryo. So you can actually, at a particular time when those PGCs are circulating through the bloodstream, you can go in and, as this will show in a minute, you can extract PGCs from an embryo. At that stage, you can culture them and you can freeze them. And so we have employed this technology. And so there you have, sorry, just before I go on, there you have in the video that PGCs, that sample of blood up into the pipetman. So if you go to the next slide, we can use those PGCs, we can culture those PGCs, we can freeze those PGCs from all of our lines, and thereby retain that genetic diversity in the freezer for future population, establishing future populations. So the process really goes something like this, where you have a broiler chicken embryo and you can take the PGCs out of here, just like I showed in the video earlier, and you can put them in the freezer. And we've established protocols for that. And we've also recreated populations from some of those frozen PGCs to illustrate the procedures. So you take those PGCs, you can culture them, you can freeze them. And then what you can do is you can put them back into this embryo that actually comes from what we call a sterile host, which is a surrogate host that we've created using gene editing technology that allows us to ablate the PGCs of the host embryo and introduce, just the same way as you saw in that video, introduce PGCs back into an embryo from the host. Now, the host is has a layer background. So layer birds produce a lot more eggs than broilers. So they give you the advantage of egg production. So if we take an embryo of this host layer, we can ablate the PGCs from the embryo and then introduce the PGCs from our donor. We then incubate that egg, we hatch the egg, and then we grow those birds into mature breeders. Now, the mature breeders look like a layer, but in fact, the gonads are from our donor, from our broiler. We mate those together, and then the offspring that we get out of those is broilers. So this is a very efficient means of re-establishing chicken populations from PGCs that have been stored. So if you go to the next slide, this is a recent proof of concept that we completed with the Roslin Institute to show proof of principle. Can we, in fact, re-establish, excuse me, can we, in fact, re-establish a line from PGCs collected from our birds? So this represents a study where we sent the Roslin Institute eggs from our broilerized silky line. A broilerized silky line has black skin, black meat, white feathers. Mike McGrew pulled out the PGCs from those embryos, and then he was able to reintroduce them into the embryos of our sterile host and introduce them into the embryo, of a sterile host, hatch those chickens, grow those chickens, and this is what you see on the left-hand side. These are the parents that are layer genetics that have gonads from our broilers, our broilerized silkies. Then we mated those two together to get the offspring, and here are our offspring, and you can see the white feathers and the black skin of those. So we can show or illustrate very nicely, without having to do any genetic or DNA profiling work, that these birds are silkies, whereas the parents have a general layer background except for their gonads. We can also look at introducing male silky broiler PGCs into hens. We can do all kinds of things so that we can ensure that all of our chromosomes are represented in future populations. If you go to the next slide, please. So this is another illustration where we've been able to expand genetic variation by gene editing some birds. So this is a parent stock. This is an inbred line held at the Roslin Institute. For a proof of concept, they gene edited the DNA of their inbred lines for the dominant white allele. And as a result, these are the offspring. So now you can see colored chicks. So there is a lot of utility of this sterile host and PGC technology for both maintaining genetic variation, storing genetic variation in our freezers, and re-establishing flocks at the other end when needed. I have to say here, just to be sure, that all of this is proof of concept work. This is all research. None of the birds created through this are allowed into the food domain, into the food chain. And so we are still doing a lot of research on this work. But suffice to say that we have also taken our lines and put them in the freezer using this technology, so we have them available for future needs. If you go into the next slide. Our conclusions are that this new technology, this PGC and surrogate host technology, or this efficient sterile host technology, has provided us an opportunity to maintain genetic diversity for future needs. We've shown on several occasions that flocks can be re-established using our sterile surrogate host. We understand that in the future, the genetics of our current commercial lines may need to be enhanced through the integration of genes from some of those zoo breeds that we have stored. And finally, we understand very well and very clearly that we have a responsibility to maintain genetic diversity or biodiversity for future poultry industry needs. And the next slide, in acknowledgement, and this is many, many years of work from the through our collaboration with the Roslin Institute. Mike McGrew leads the efforts there, but there are a range of other people at the Roslin Institute that have participated in this study, and the various proof of concepts that we have completed with the Roslin Institute. And we're very grateful that we've been able to create this tool to allow poultry production to be efficient in the future. And that's the end of my talk. I'm happy to take questions at some point. Yeah, I see some questions in the Q&A, and I will attempt to answer those as we can.
Yes, thank you very much, Rachel. To the next presentation from Pierrick. I hope we can see him somewhere, and he's we can hear you. Pierrick, are you there somewhere? Okay. So Pierrick works in SYSAAF, another member of EFFAB. SYSAAF is an umbrella organization representing animal breeders in France, active in poultry, aquaculture, and insect breeding, if I'm not mistaken. And Pierrick has a strong background in animal genetics and design breeding programs, and also in phenotyping tools and yeah, many other things. I'm not going to take more the floor, and I give you. Hope you can share your presentation.
So thank you, Anna, for this introduction. I'm managing aquaculture unit in SYSAAF, which is French Association of Poultry and Aquaculture Breeders, and since last year also insect. And I will give you some example of what has been done with the French breeding companies in trying to maintain or to create genetic diversity. First, a short presentation of SYSAAF, excuse me. SYSAAF, is French Poultry and Aquaculture Breeders Association. It's a non-profit organization. It's a pool of 27 geneticists and data scientists working for 31 breeding companies, rearing and selecting more than 30 different species, among some of them in aquaculture. We are doing a genetic indexation of more than 100 lines and providing some expertise, mostly in aquaculture in different countries, Thailand, Mozambique, and Mexico, and Madagascar. You have here the company which are members of poultry breeding of our union, here in trout breeding, in marine fish breedings, for production of caviar and fresh waters, some shellfish, and in shrimp breeding here. And since last year, some companies were desiring to develop breeding program for insects. So coming to the talk and the subject of the talk, aquaculture has just initiated domestication. And one of the main problems of aquaculture is may not be to maintain, but how to create well genetic resources. Here you have a figure of the number of species which were domesticated in land animals, in plants. But in aquaculture, we can see that it's a recent event in the last centuries, and most of them in the last years, last decades. So the challenge for aquaculture is only to maintain, but also to create well genetic resources. And there is a document from FAO, which is really interesting if you are interested in the subject, to see and to read what are the questions around today and how to go in this direction to create new genetic resources. So my core message in this talk is that aquaculture nowadays are actively involved in the management of genetic diversity. I will provide example in characterization of genetic variability of their stock, because in many cases, there is no information. In using tools to trace pedigree, using family relationship, in using, for example, DNA parentage assignment or fingerprinting, in improving or adapting softwares to this new activity of using DNA to know who is who in stock, in applying genetic principle to optimize conservation of genetic diversity. I'll give you an example. And I will finish by, excuse me, use of cryopreservation for long-term private and public conservation of genetic resources. First example is about evaluation of genetic variability for successful domestication of species. I have taken here the Mair, Argyrosomus regius, which is rare in Mediterranean area. This species is reproducing in only few areas worldwide. And we collect tissue samples for genotyping with microsatellites in these different countries, in Mauritania, in wild animals, in Egypt. And for this project, we were to get some agreement with Egyptian government agency for defining access and benefit sharing only for the samples. And results from this study were to see that the different populations are very differentiated. Here you have the FSA differentiation of the population. You can see that all populations are differently, but some of them from Turkey, Egypt to the Atlantic ones are much more differentiated, probably near at interspecific level somehow in some way. This study also allows us to evaluate genetic diversity, is published now, and it allows us also to evaluate genetic diversity's heterozygosity from the wild population we collect in the wild from Egypt, from Mauritania, Portugal, Turkey, and from hatchery population in France. And we can see here that the hatchery populations that were collected have nearly the same variability as the wild population. So it's the first basis and good basis to initiate breeding program. I will now go to why to take care, even when you create genetic resources, to take care of inbreeding. Here it's a short, limited presentation of potential risks. To not take care to cross brother and sister. It's an old study from Gallardo in rainbow trout, where they crossed during 3 generations brother and sisters, and you can see that fry survivor here, the body weight at commercial weight, and then the productivity when you multiply the two are going down very quickly. You don't take care about that. So there is also some negative effect, additional negative effect as malformation and sensitivity that have been reported in the literature. So there is no long-term sustainable breeding program without management inbreeding during domestication. What is a breeding program in aquaculture? Typical breeding program is to create a lot of families. Each family is the progeny from one dam and one side in one incubator. And then you, as the fish or the progeny are very small, you are obliged to rear them in one tank per family until you are able to tag them with electronic transponder, then to mix them at 50 grams, for example, and to re-rear them in a different cage or tank or raceway, and to measure and to do selection, to measure different kind of traits. And then we use same kind of tools, softwares and genetic softwares and in livestock, doing genetic and genomic indexation to propose the best candidates, maximizing genetic progress and minimizing inbreeding, the balance of the subject of today. So, how to get pedigree in aquaculture? So you can see the size here of the animals that are hatching are very small. So this is justifying the fact to get one tank per family. This is need an investment in facilities, in personnel, which are very, very large, very, very expensive. Another way is to pool the families together that are hatching or fertilization and to rear them in only one tank, one environment. And you have to do a posteriori DNA fingerprinting. You need to collect DNA from the fin. You have to tag the individuals with a transponder and to genotype them to know who is who. In this case, this kind of other kind of breeding program, there is no specific facilities which are needed, so no initial investment before return rate of investment, but you have genotyping costs. And there is no or limited within tank effect because you have only, they are all in the same tank. So it makes that more precise. That's why in France it was favorized as approach, as more adapted to SMEs. So we introduced DNA fingerprinting in shellfish breeding here in Pacific oyster, a panel for parentage assignment to know who was developed with EFFAB using SNP markers, which is different at 1 bare base in chromosomes, and using more than 100 SNPs markers. And this was published, for example, in 2014, so it's really affordable. And we test this panel in different private companies, hatcheries, and different kind of design to see if when they are doing matting for new next generation, they will increase or create in breeding or not, or increasing breeding. Here the 4 companies were Satmar, Marinov, Novostria, and Vandenessa. And they made, for example, here 3 dams with 7 sites, creating 21 families, 4 times, more than 80 families. And 2 years later, we see after genotype animals and to see after who is who and what is the remaining diversity. And here you have coefficient of inbreeding. And the matting design was different according to different hatchery practices. And we can see here that even with 80 families created here, the rate of inbreeding observed, coefficient of inbreeding was nearly 3%, which was too high. Even if this was in 1 year class, it has to get 2 year class for 1 generation of oysters. You can see here also is 2%, and we estimate it's too high. This is without using fingerprinting. When using fingerprinting, you know who is who, you're able to choose and to equilibrate families, then inbreeding or coefficient of inbreeding is going down in all the cases. So now this tool was used during 2 generations by breeding companies. It helped to secure domestication in managing inbreeding risks. Second example I will take you is in shrimp breeding. In France, the blue shrimp, here it's really blue, was introduced in New Caledonia in the 70s with a limited effective of founders, probably 3 to 5. It was an initiation of aquaculture. And since this time in the 90s, there is a failure to introduce new genetic resources from source not resistant to the virus. So in the last years, we developed a panel of more than 100 SNPs identified by rat sequencing. And here you have the variability of the different pool of individuals that were sequenced from different hatcheries in New Caledonia, and compared to the variability of samples from wild in Mexico. You can see the variability is lower. And we use that in a breeding program to try to see if we are able to test and to trace pedigree, but also if there is remaining genetic variability with a so low founders numbers. And we estimate heritability, for example, for different traits, body weight, length, color, on raw shrimp or cooked shrimp, which I think to look for red shrimp. And you can see that there is a large remaining variability, even the founders is limited. And this was done in more than 1,000 progenies from more than 40 families. The conclusions were that there is, even with limited genetic variability, it is not preventing the future genetic progress. And regarding the difficulty to introduce new source or new resources, the management of the genetic resource locally was proposed and is planned now. Similar démarche is in progress in Polynesia also. So, parentage assignment became widely applied in SYSAAF here in aquaculture. For example, we go to achieve more than 40,000 shrimp individuals, genotypes per year. It's routine for the breeding companies I present you initially. So I will go to another application or improvement or application of breeders is to adapt software to assign parentage assignment to parentage in an octoploid species. In France, we are producing caviar from sturgeon, Acipin cerberi, which was introduced from Russia in 2 times with potentially very limited genetic variability. But this is one problem. The other problem or the other difficulty when doing assignment, for example, in human, you have the genotype of the progeny of the people, and you want to see the mother and dams and sire with who. You have only two possibilities coming from father and the dams. When the species is octoploids, you have four possibilities of alleles or variability associated with each parent, making the usual software not adapted to this case. So within our laboratory, we adapt Vitessine software, which is software to do assignment, to octoploid level, under two very simple hypotheses: that all variability present in an offspring should be present in its parents, which is logical, and with applaudi N, the parent should not need to bring more than 50% of the allele to be true. Regarding that, it was very challenging to know if it's possible. We test that on a crossing done by a student company. They have the dams, the males, the dams, and the families created in two cohorts. And then we estimate heritability of different production traits, here using nearly 500 female genotypes and phenotypes, and genotypes with microsatellite markers. You can see, even with very limited, probably, introduction, we were able to genotype most of the animals. Only two females were not assigned. And there is still remaining, and there is high interest in variability for body weight, oocyte size, which is interesting, caviar yields, or caviar color. And it means that it is possible to DNA assign in commercial condition and octoploids animals to improve stock and manage inbreeding. And this was published this year. So, two last examples of improvement of implication of breeders is how to mate, is to an optimization of mating to minimize increase of inbreeding. So the question for the breeders is how to choose who to mate with who to create a new generation. And we apply.
Pierrick,
Yes.
can you please go a bit faster, because we are taking a lot of time of the panel discussion. I'm sorry.
Yes. Okay.
Thank you.
Example. Thank you, Anna, for reminding that. So we use application of optimal contribution selection adapted to poultry and aquaculture in SYSAAF in 2010-12. We test use of this principle on 3 commercial population, 3 breeding programs, using evaluation of linkage disequilibrium of part of the chromosomes which are homozygous and very long. And here it was published by Jonathan D'Ambrosio last year and previous year. You have here what was the decrease of effective size of the population, even with taking really care about inbreeding. And then the drop, the increase with introduction of optimal contribution principle in the management of the breeding program. This means that combining here fingerprinting and optimal contributions, you are able to increase and to improve management of the conservation of the genetic resource within line. The last example, excuse me, Anna, is also related with KIP discussion, is for long-term conservation of genetics using cryopreservation. We initially worked with INRA and IMV developing extenders and conservation procedures, and then with the Evolution Breeding Company in cattle, EFFAB, INRA, and Cryobank National, we set up a collective cryobank, CryoAqua, which is operational since 2012. In this area, we have a specific area for frozen to freeze sperm or eggs for aquaculture species, and each breeding company has its own containers with their own security and sanitary take care. And you have here what was frozen different species from the breeding companies. Here you have the number of males which were frozen, and here with the lines, the straw number, which were frozen since the last 10 years, less 10 years. This is for private usage, and the same CryoAqua cryobank also is doing cryopreservation for in public cryobanking from the same animals provided by the breeders. And here you have the number of males that were frozen and straw produced and in order to provide to Cryobank National within the CRB project, which is to store animal genetic resources, livestock. So spray and cryopreservation is now routinely used and operated to conserve genetic resources by the breeders, and the partnership with the Cryobank is securing long-term conservation of genetic resources. I think it was interesting to show this kind of example today for discussion. I will stop my my presentation now. I want to thank you for your attention, and I give the speech to Anna and the floor.
Thank you very much, Pierrick. So I'll give the floor to Chala. Chala, where are you?
Well, welcome all to our panel discussion, and thank you all for accepting the invitation as well. So we have three invited panelists. We have Graham Mair, the Senior Aquaculture Officer from the FAO Fisheries Division. So Graham, I can't see you. If you're here, can you open your, raise the hand. Graham raised his hand, so let
Graham
me see.
was here.
Okay, Graham, you are now able to talk. Please, unmute and unmute. So the floor is yours, or maybe until you solve the microphone problem, I can move on to Juan, María Gallardo from the Spanish Federation of Livestock Purebred Associations. Hello, Juan. Can you shortly introduce yourself and your organization?
Yeah. Hi, good morning. I'm Juan María Gallardo. I'm from FEAGAS, like you told in the short form. We have a purebred associations federation. We have 114 associations of different kind of animals, cattle, sheep, goats, and horses, pigs, and so on. And I'm a veterinarian, and I have the PhD too. So I'm here to solve all the questions that the people ask.
Thank you very much. It's very nice to have you here as someone from the field, the farm level. So I can now give you the floor, Graham.
Thank you very much, and thank you for inviting me to join this panel. So my name is Graham Mair. I'm a senior fisheries officer or aquaculture officer with FAO, but my principal responsibility is to work on our programs on aquatic genetic resources. Paul's introduction gave a very useful concept of how FAO goes about looking at genetic resources from a global perspective. We are doing a similar thing on aquatic genetic resources, but we are far, far behind. And one of our major goals is to try to elevate and try to build work on aquatic genetic resources. So we have published the State of the World report. We are now developing information system on aquatic genetic resources, and also we now have a draft global plan of action. So we hope that all of these instruments will facilitate more work and better, because often aquatic resources get left out of the discussion on diversity management, and we want to try to make sure that happens less often. So nice to be here.
Yes, and thank you for pointing that out. I will get back to you in a minute, but first I would like to also introduce our last panel in IT. Waltraud Kugler is from SAFE Foundation. And can you shortly, briefly introduce your organization, Waltraud?
Yes, I'm from SAFE Foundation. SAFE Foundation is an abbreviation of Safeguard for Agricultural Varieties in Europe. We are started in 1993, so we exist nearly since 28 years. Our focus is pan-European network to connect organizations which strive to hold the reverse and the trend of genetic erosion in agrobiodiversity. We act as a representative of NGOs and farmers and breeders. And our structure is we have European-wide about 25 partner organizations in 16 countries and 2 regional organizations. We are funded under Dutch law. We have a network office in Wageningen and a project office here in St. Gall, where I'm speaking from. And for funding reasons, we have 2 associations in Germany and in Switzerland. That is SAFE Foundation.
Perfect. Thank you very much. So you will all be invited to also respond to the questions, even though they are sometimes directed to the speakers. So let me start with the first question. This was for you, Sipke, but I think there are others that would like to respond to this as well. How do you intend to balance maintaining biodiversity in an era whereby animal production is profit-driven and farmers would rather use high-quality genetics like Holsteins? I think this is the question which everybody has in their mind.
Yeah, well, thank you. I think it's the key question of this whole webinar. So farmers choose the genetic resources, the material that they need. But from a breeding perspective, I think there's really a commercial interest, also a long-term commercial interest to maintain genetic diversity while at the same time making genetic improvement, as was presented by Rachel also. So it's a long-term, short-term balance, I would say. And if you look a bit in the future, long-term, then you should also look into maintaining genetic diversity in your breeding program.
Yeah. Juan, would you like to also respond to this as a national organization?
Yeah, I think that it's a difficult balance too in the long-term, short-term process too. So I don't have any more.
Okay.
For us, it's too complicated to say to the breeders that they need to not only to collect the samples to commercialize, not only to frozen and save them. But it's a difficult wait for us.
Okay, thank you. Waltraud, you raised your hand.
Yes, I think we have experience over the last 28 years to see how genetic diversity or different breeds were accepted through the farming sector, but also through the public. And I think a change is going to take place now. We see this with our European ARCANet, where we have farmers in, where the public can visit and can have a look at. That is one point where I completely agree with Sipke that there is an increasing interest, and also the industrial interest is increasing because of big inbreeding problems with the main breeds, especially in the cattle sector. And the other thing is that we have really to look closer to environmental issues and also to the consumers' interests. And so I think the main point is to motivate the farmers and hobby breeders to keep their breeds clean.
Thank you, Waltraud. Any other comments on the topic?
There were 2 questions in the QA box related to this topic that Waltraud was raising on the trade-offs, perhaps on the environmental impact of animal farming in Europe. Can you perhaps? And yeah, and Graham has raised hand. Sorry, I'm sorry. I was checking your role. Sorry. Go ahead, Chara.
That's okay. I just wanted to make a very quick comment in relation to the discussion just now. I won't comment in relation to aquatic genetic resources because the system is, we are in a different place at the moment and we don't have so many developed breeds. But I think communication is a vitally important tool here. And I think communicating the needs, both in terms of the negatives if you don't have them, but also identifying case studies of where these older breeds have been conserved and have actually been utilized to the benefit of the industry, because I think it's not enough just to say that it's a good idea. It has to be communicated very, very effectively on what is the benefit, including economic benefit of these breeds. Thank you.
And Graham, you already had a question yourself to Sipke about having aqua species included. Maybe you can directly ask it.
Okay, it's my panelist privilege, I guess.
Yes.
Yes, Sipke, in your talk, you made a reference to aquaculture, but you were referencing plants, animals, and forest genetic resources, and generally speaking, those tend to exclude aquatic species. So I'm just wondering if there is a plan for either to involve aquatic species in the existing strategies for animals and plants, because obviously they are animals and plants, or whether there's a plan for another strategy that can focus more on these aquatic resources within generous speech.
Yeah, thank you. It's a very good question, and we also discussed the scope of the strategy for several times, and we started from a request from the European Commission on the 3 networks on plant, animal, and forest genetic resources, the existing networks in Europe, to develop this strategy for plant, animal, and forest genetic resources. But what we have in mind when we have this integrated strategy, that we should be able to expand it to other genetic resources also in the future, like aquatic, but there are also other genetic resources, microbial or whatever. And if you see the council recommendation or decision of the farm to fork strategy, there was indeed a reference already made by the European Union to include aquatic also. So I can imagine that aquatic genetic resources will be included sooner or later.
Yeah, and I think some of the tools and resources that FAO are developing should facilitate that.
Yeah, exactly. That's what we have, as Sipke said, we were discussing this within Genrich Bridge project, and there is a very important thing. So yeah, I agree with you, Graham. I already made this question to the group.
I stand ready to help.
Okay.
So in terms of the environmental impact of the farmed animals in Europe, there was a question that doesn't highly genetically selected animals have less overall environmental impact? So would it be worthwhile to focus on improving the environmental or mitigating the environmental impact of the traditional breeds and understanding their genetic makeup while they're being biobanked?
I think the question was for me, right?
Yes.
Yeah, it's an interesting question. I think in general, we think that high productive, high input, high output breeds are most efficient in terms of per kilogram product. But if you look at it in a different way, more at farm level or landscape level or global level, and you take into account all costs and all values, then different breeds may be more optimal in certain production system or production environments. So I agree with Mike that we should continue to select animals to be more efficient, but it really depends on the system and the requirements on the system. What is the most optimal genetic product, I would say, or breed.
Thank you, Sipke. Any other comments are welcome.
Pierrick has raised his hand. I don't know it's about that.
Yes, on the same subject, we have done some estimation of life cycle analysis, considering traits to be improved and to reduce environmental impact. And we began by fillet yield, for example, which is interesting traits, because when you calculate related to the meat consumed by people, not by ton of produce or fish produce, you have a very important impact, because a major impact is feed in aquaculture, in environment, and if you increase or limit improve feed efficiency, you have a very important impact. So the one aspect is are the bred lines to be selected. The other are the system of productions. And they have not to be opposed, and a good line in the system can be used also. So it's more complex probably than to say or to limit only lines, the problem to only lines.
Yep. And Chala, we cannot hear you.
And maybe just another related question, that how do you see as the key incentives? What do you see as the key incentives for cattle, pig, and breeding companies to support the endangered breeds?
The question was directed to me, but I would also like to forward the question to our Spanish colleague, but let me first try to answer myself. I think well, breeding companies, of course, they should look at current and future production systems. And what we see is that things are changing depending on what kind of requirements and threats like climate and biodiversity threats, environmental pressure are being put on production systems. So I can imagine that cattle and also pig breeding companies look at a larger product portfolio towards the future, including a broader range of breeds that they would like to maintain or market. And secondly, I think there's also some societal responsibility maybe for these companies to help supporting the conservation of really endangered breeds.
Yeah, that's perhaps Rachel she wanted to jump before, and we're talking about breeding companies, and before Juan, María.
Yeah, I just wanted to say that, you know, taking some of these traditional breeds or non-commercial breeds as they're being biobanked, it is important to understand the genetics or the genetic makeup of those breeds, and more importantly, what are the genetic elements that are resulting in the expression of a particular phenotype. I think that's the real key, is exploring the genetics that we're biobanking, exploring the genetics of those diverse lines, diverse breeds. Because when it comes to introducing new performance metrics or new characteristics into commercial lines, we will end up having to use integration technologies or technologies like gene editing to get those genetics in. So I think it's very important that we do explore those genetics. Now, upon saying that, I know that as a breeding company, we don't have the resources to explore the genetics of all of our zoo lines. I wish we did. I wish I could understand exactly what are the genetic elements that are creating the new performance phenotypes, or the different performance phenotypes that we see in these different breeds. But I do think it's a very worthwhile activity. It's just a very expensive activity.
Yeah. Juan María, would you like to comment?
Yes, I only want to comment that we don't have close treaty with the big genetic companies, that we also have the association of a breeds, not that big company. So we don't know in the field that what's the kind of these companies that they have. Rachel or Pierrick maybe could apport something more.
This discussion, the fact is that genetic improvement is mostly driven by the market, and but there are some new markets, as was said by Waltraud just at the beginning of the discussions, and there are many examples in Europe where you can associate the genetic product and the final product for the consumers, making the consumers able to pay more for some kind of product. So this kind of approach relies, for example, in some cheese in France or chicken also. And the final product is strictly associated with the genetic component, the lines, initial line, traditional lines. And this kind of way to make the cost supported by the market is another way to say the role of the farmers and the breeders. And I don't think we have to oppose the large breeding company and the other one. It's only perhaps that today we have to create the conditions to make the cost paid by somebody.
Yeah, that's the important part, I think.
Exactly.
There was a question for Rachel, so I want to ask that. Can you tell us about the success rate of the PGC injection, like how many donor embryos needed to get a full-grown mature adult, and have the genotypes produced through these been tested for adaptability outside the experimentally designed environment?
Yeah, look, I'd have to go back to the paper to look at the statistics of how many injections was required. I don't have that number at the top of my head, I'm sorry. These populations are only being grown in the experimental house. We certainly haven't had permission to take them out into any of our production systems at this point. But they are just the genetics of the line, really. When you look at, you're freezing a primordial germ cell, which is a diploid cell. You're putting in a combination of those PGCs back into a single animal and breeding. I mean, it's just regular breeding. There's nothing special about that except the surrogate itself. And the surrogate was only engineered so that we could get rid of the host primordial germ cells so that our broiler germ cells could have a chance of being maintained. So I don't see that there would be any impact. The birds are healthy, they survive they grow as we expected, they survive as we expected. So I don't see that there will be any issues.
Thanks, Rachel. I think we have to stop by here. I'm so sorry, but we have to stay a bit on the frame we have designed at the beginning and respect the time of our participants. So, Pascale, can you perhaps make the conclusions as was expected?
Yes, with pleasure. Thank you, Anna. And thank you to everybody, because that was a very interesting session. So having FAO monitoring, assessing the worldwide diversity, that demonstrates that the genetic diversity is totally linked with the worldwide food security. And it's interesting to listen to the EU genetic biodiversity setup story by Spike. Good to hear as a fact that genetic resources are key components for innovation. Actually, we need to preserve the genetic diversity and use it to go through all transitions. Agriculture has to go on entering year after year. We heard now how Cobb identifies lines of interest, conserve lines, germ cells, very interesting technology, and is able to reestablish a line of interest. Pierrick, the younger of our animals branches involved in domestication, listed some of the key factors of success for the genetic diversity management, talking between others genotyping, about DNA fingerprinting, and also, of course, data and softwares, which are very important for our genetic management. Pierrick showed us also that a good management of the genetic diversity helps to maintain the required diversity even within small populations. And myself, as a general manager of the French National Union of Breeding Companies involved in ruminants, ALICE, we are involved in ruminant semen and embryos dissemination. I would add that in ruminant species, we also monitor the inbreeding dynamics and develop tools that the breeders and the technicians can use to monitor consanguinity levels. As I have the floor, I can also be a witness. There was a question about the conflict between economic interest and genetic selection. Just to say that the new trends in farmer choices is not anymore to have Holstein produce more and more milk, but to have resilient animals in good health, adapted to the climate change, and with less gas emission also. That's the new traits and the current traits on which the selection is going on. And before finishing, just to say at last, actually, for all of us involved in genetic development, that we have designed a code FABRE in EFFAB, which has a pillar dedicated to the genetic diversity. And in this genetic diversity management pillar, of course, we talk about how to store, keep in-house breeding population, how to store the genetic resources we need to have for the future, and also how to monitor that we engage ourselves also to monitor the inbreeding rate and make sure that we don't lose the genetic resources when we develop our economic models. So thank you to everyone. Thank you to the speakers. Thank you also to our three panelists who accepted to play the role, to answer the questions and take part in the animation. And of course, thank you to the very great EFFAB team led by Anna. Thank you for this fourth session of this Breeders for Green initiative, which is a very great one. Thank you to the team. Bye to everyone.
Yeah, thank you everyone. Thank you, Pascale, for the conclusions. Thank you very much, Chala, for moderating this short panel discussion. Sorry, because the topic is, interesting. Looking forward to the publication of the strategy in November 2021, SIPCA. Hope we can make an in-person event here in Brussels, and we keep you updated about it. Stay tuned. We will come back in the autumn with another session on the breeder stock green. Bye-bye.
You. Bye-bye.